Today, I want to start with a bit of a thought experiment for you listening. Imagine something that has no brain, no stomach, no lungs, and absolutely no ability to move on its own.
Right.
It's sitting on your desk right now. Maybe it's on the surface of your coffee cup. And to the naked eye, and honestly, even to a standard lab microscope, it's completely invisible.
Yeah, just a
Ghost. Exactly. It is, for all intents and purposes, just a complex speck of dust, dead matter. But if you touch that coffee cup, and then you touch your eye or your mouth, that speck of dust wakes up.
But it doesn't just wake up.
No, it takes over. It completely hijacks the machinery of your cells and forces them to build an army.
It really is the ultimate sleeper agent, isn't it?
It really is. So today we're doing a massive deep dive into the general properties of viruses. And I have to be honest, before reading through this source material, I thought I knew what a virus was.
Most people do, yeah.
I thought, okay, a germ, it makes you sick, you take some vitamin C and rest. But looking at the research, specifically this chapter we're unpacking today on viral properties, the reality is just, it's so much stranger. The text describes them literally as parasites at the genetic level.
And that phrase, parasites at the genetic level, that is the absolute key to this whole discussion.
Right.
It shifts the perspective immediately. We aren't talking about a bug that eats your cells like some microscopic predator. We are talking about a piece of code, a set of instructions that infiltrates your own operating system.
But let's back up a second and establish the physical reality here. Because the very first thing that hit me in the text was the scale. We throw around words like microscopic all the time. But viruses are in a completely different league.
Oh, entirely.
The source material establishes they are the smallest infectious agents, ranging from 20 to 300 nanometers. And I just have no frame of reference for that. Help me visualize 20 nanometers.
Okay, let's try this. You know what a bacteria looks like generally, like a staphylococcus.
Sure, like little round blobs if you look under a microscope.
Right. So a staphylococcus bacteria is about 1,000 nanometers in diameter. Now compare that to a human red blood cell, which is huge by comparison, roughly 6,000 to 8,000 nanometers.
Okay.
Now take a small virus, like the polio virus. It's about 20 to 30 nanometers.
So if the red blood cell is a stadium...
If the red blood cell is a massive football stadium, the bacteria is maybe a bus parked down on the field. The virus is a marble in the pocket of someone sitting way up in the nosebleed section.
Wow. Okay, that really puts it in perspective. We are talking about something so incredibly small it can barely contain its own existence.
Precisely. And that size limitation, it dictates everything about them. Because they are that small, they simply can't carry luggage.
Right.
They can't have a metabolism. They can't have ribosomes to make their own proteins. They are stripped down to the absolute bare biological essentials.
And this leads to that core paradox I mentioned in the intro. The text highlights this right at the start. They are totally inert in the extracellular environment. This kind of blows my mind. If I have a vial of, say, smallpox and I set it on the table, it's not doing anything.
Nothing. Yeah. Zero biological activity. It's not consuming oxygen. It's not creating heat. It is, chemically speaking, a complex crystal. Yeah. It's just an assembly of macromolecules.
Wow.
And this is exactly why the debate rages on among scientists. Are they actually alive?
Well, are they?
Well, it depends on your definition, really. If life requires independent metabolism, eating, generating energy, excreting waste, then no, they are absolutely not alive.
Right, because they can't do any of that on the table.
Exactly. But if life is defined purely by the ability to replicate and evolve over time, then yes, they are. The text calls them obligate intracellular parasites. They only live when they are actively inside you.
So they're basically vampires. They need an invitation to enter your house, and then they drain you.
A vampire is a decent analogy, yeah, but a vampire still walks around and hunts on its own. A virus is honestly more like a thumb drive you find in a parking lot.
Oh, that's good.
It does nothing. It's just plastic and metal until you plug it into a computer. Then it executes its code.
That is terrifyingly accurate. Okay, let's look at the hardware of this thumb drive then. The text makes a really specific distinction early on between the virus as a general concept and the virion. What is the difference there?
It's a technical distinction, but a super important one for the literature. The word virus can refer to the infected cell, the entire replication process, just the whole phenomenon. But the virion is the physical particle itself.
The actual object.
Right. It's the delivery truck. It is the fully assembled infectious unit containing the genome and the shell, ready to go out and infect the next cell.
So when we sneeze, We aren't just spraying virus. We are specifically spraying virions.
Exactly. Millions of them.
All right, let's dismantle one of these virions. Our mission today is to unpack every line of this text. So let's start with section 1, the anatomy lesson. If we are building a virus from scratch, what do we need?
Okay, at the core, you have the treasure, the nucleic acid. This is the genome, the actual genetic information. And the text notes a strict rule here. It's either DNA or RNA. It is never both.
Never both, got it.
Right. But you can't just have naked genetic material floating around in the wild. It would get shredded by UV light or host enzymes instantly. So you have to build a protective box. That box is called the capsid.
The capsid. And this is a shell made of protein.
Yes, it's a protein shell enclosing the genome. Its job is twofold, really. Protection, obviously, but also delivery. For many viruses, the capsimen itself is the thing that recognizes and attaches to the victim cell.
Now, the text gets a bit into the weeds here with terms. It contrasts capsimers with structural units or protomers. I read this section a few times, and it felt a bit like splitting hairs. Why did this distinction matter?
I get why it feels that way, but it matters because of the difference between how we observe them under a microscope versus how they are chemically built.
Okay, break that down for me.
Imagine you are looking at a brick wall from a distance. You see a pattern, maybe a distinct diamond shape formed by the bricks. That visual pattern you see, that's the capsimer. The text calls it the morphologic unit. It's the shape visible under an electron microscope.
Okay, so the capsimer is just the visual pattern.
Right. But if you walk right up to that wall and touch it, you realize that diamond shape isn't one solid thing. It's actually made of 5 separate individual bricks glued together.
Ah.
Those individual bricks are the structural units, or the protomers. They're the actual basic protein building blocks. And a subunit is just a single folded viral polypeptide chain. So the text is basically warning us. Don't assume the bumps you see on the surface of a virus are single massive proteins. They're usually clusters of smaller proteins.
Got it. So the protomer is the chemical reality, the actual protein, and the capsimer is just the visual shape it forms.
Exactly. And when you take the nucleic acid and you put it inside this protein capsid, we call that whole assembled package the nucleocapsid. For a lot of viruses, that's the whole virion. They're done. We call those naked viruses.
But some of them decide to put on a coat. We need to talk about the wrapper or the envelope?
The envelope. This is a massive game changer for viruses. Some viruses surround their nucleocapsid with a lipid-containing membrane.
And where exactly do they get a lipid membrane? We just established they're too small to make anything like lipids, right?
Yeah, they can't synthesize it. They steal it.
Identity theft.
It is the ultimate identity theft. When the virus is finishing up and leaving the host cell, instead of just bursting through the wall, it gently pushes against the host cell's outer membrane and wraps itself in a piece of it. It buds off. So the outer surface of the virus is actually made of the host's own skin, essentially.
It sounds like an absolutely perfect disguise. If it's wrapped in my own cell membrane, does my immune system even recognize it as foreign?
That is the whole idea. It looks like self. However, the virus isn't stupid. It knows it needs to attach to new cells eventually, and a plain, smooth lipid bubble won't stick to anything. So just before it buds out, it inserts its own specific proteins into that patch of host membrane.
The peplomers.
Yes, the peplomers. Or, as we hear much more commonly in the news, the spikes.
Right, the spikes.
If you picture the classic image of the coronavirus, those red petal-shaped spikes sticking out of the gray ball, those are peplomers. They are virus-encoded glycoproteins exposed on the surface. So it's wearing a stolen coat, but the virus has pinned its own functional badges all over it.
And those badges, those spikes, are what our immune system eventually learns to target, right?
Exactly. Those spikes are necessary for the virus to infect the next cell, but they are also its Achilles heel. They are the most exposed part for our antibodies to grab onto.
Before we move on to how we classify all these, I found one definition in Section 1 strangely sad. The defective virus. Sounds like a toy that just came out of the factory broken.
It's a very common phenomenon, actually. A defective virus is a particle that is functionally deficient in some aspect of replication.
How does that happen?
Well, maybe it packaged its DNA wrong, or it mutated and is missing a crucial gene for the capsid. It can physically enter a cell, but once inside, it just stalls out. It can't reproduce.
That's just a dead end. It dies there.
Usually, yes. Unless, and this is wild, it gets lucky. If a cell happens to be infected with a defective virus and a normal, fully functional virus at the exact same time, the normal one acts as a helper virus.
Oh, wow.
It provides the missing parts. The defective virus basically mooches off the helper's machinery to replicate itself anyway.
So it's literally a parasite of a parasite.
It is parasites all the way down.
Okay, we know what the hardware looks like now.
Yeah.
But where did these things even come from? Section 2 of the outline tackles their evolutionary origins, and it presents two main theories that are radically different from each other.
This is one of the great enduring debates in biology. We don't have a fossil record for viruses. They don't leave bones. So we have to guess their history based purely on their genetics and structure.
Right. So theory number 1, cellular origin.
This is often called the escaped gene theory. The idea here is that viruses started out as just normal, functional pieces of DNA or RNA inside regular host cells, maybe a piece of messenger RNA or a gene for a specific protein. And somehow, through a genetic accident, these pieces gained the ability to replicate on their own, acquired a protein coat, and gained autonomy. They essentially went rogue.
I love this idea. It's like a worker in a massive factory just stealing the blueprints and setting up their own competing shop right across the street.
That's a great way to put it. And the genetic evidence really supports this for some viruses. When we sequence them, we find viral DNA sequences that look suspiciously similar to normal host cell DNA. It strongly suggests they are our own wayward genetic children.
But then the text gives us theory number 2, regressive evolution.
This is the fallen angel theory. The idea here is that viruses used to be much more complex. They might have originally been small, independent cells, or maybe bacteria that started parasitizing larger cells.
And then they just got lazy.
Basically, yes. Over millions of years, they realized they didn't need to expend energy making their own food or building their own structures because the host cell provided all of that for free. So evolutionarily, they shed the dead weight, they lost their stomach, they lost their locomotor system, they lost their cytoplasm. They devolved until they were nothing but a naked shell and a genome.
Ruthless efficiency.
Exactly. Why carry around a heavy gene for metabolism if you can just steal the host's ATP?
Right.
But the text does note a caveat here. While this regressive process absolutely happened with certain parasitic bacteria like Rickettsia, we don't have hard, concrete evidence linking viruses directly to bacteria in this way.
Except maybe the giant ones.
Yes, exactly, the poxoviruses. They are so massive and complex that some virologists think they might actually be the remnants of a cellular ancestor. But for the tiny viruses, the escape gene theory is generally more accepted.
It's highly likely both theories are true, right? Just for different viruses.
Almost certainly. Viruses are polyphyletic. That means they don't all trace back to one single common viral ancestor. They have many different origins. They aren't one big family tree. They are a diverse collection of different genetic entities that all just happen to land on the exact same survival strategy.
Speaking of families, let's get into how we classify them. The text outlines the taxonomy. We have families ending in Viridae, genera ending in virus, and the scale is huge. Over 4,400 species across 122 families.
It's a massive catalog.
And they classify them using 7 specific criteria. Morphology, genome properties, genome organization, protein properties, antigenic properties, physicochemical properties, and biologic properties. I want to do a sort of roll call based on this. We obviously can't hit all 4,400, but I want to survey the major families that affect humans.
Let's do it.
And I don't want to just read a list. I want to know their personality. If these viruses were characters in a movie, who are they?
I like that approach. We categorize them first and foremost by their operating system. Are they running on DNA or are they running on RNA?
Let's start with Section 3 of the DNA lineup. These feel to me like the establishment. They are usually more stable, right?
Yes. Chemically, DNA is a very robust, stable molecule. These viruses tend to be consistent, their mutations are slower, and many of them have evolved to hang around in the host for a very long time.
First up on the roll call, Parvoviridae.
The runt of the litter. Parvis literally means small. These are the minimalists. They are incredibly simple, only 18 to 26 nanometers, and because they are so tiny, they have a minuscule genome made of single-stranded DNA.
Single-stranded DNA.
Yeah, which is unusual for DNA. They literally don't have enough genetic code to even start their own replication process. They have to sit and wait until the host cell naturally decides to divide, and then they hijack the cell's replication machinery while it's active.
So they are completely opportunistic.
Completely. The big pathogen here for humans is the B19 virus. It causes fifth disease in kids, that bright red slap cheek rash. It's usually mild, but because it specifically targets red blood cell precursors, it can be really dangerous for people who already suffer from anemia.
Okay, next we have 2 that seem to go together in my head, Anelloviridae and Polyomaviridae.
Let's take Anelloviridae first. Anello means ring. They have a circular genome. They are fascinating because they cause chronic, lifelong infections in a huge percentage of the human population, but, and this is weird, we haven't linked them to any specific disease yet. They're just kind of along for the ride.
Wow, just silent passengers. And Polyomaviridae.
These are the saboteurs. Poly means many, and oma means tumor. They can produce tumors in infected hosts. The JC virus is in this family, which causes a severe brain infection called PML in immunocompromised people, and the BK virus, which is a major issue for kidney transplant patients.
And closely related to that concept is Papillomaviridae.
Right. Papilla means nipple, which refers to the raised wart-like lesions they cause. This is HPV, the human papillomavirus. And like the polyomaviruses, they are saboteurs. The high-risk strands of HPV don't just make warts. They can accidentally integrate their DNA into the host's chromosome and basically break the cell's brakes.
Leading to unregulated growth.
Exactly, leading to cervical and other genital cancers.
Next is Adenoviridae.
The common thug. Named after adenoles, meaning gland or lymphoid tissue, because they were first isolated from human adenoids. These are tough, naked, non-enveloped viruses. They cause your standard miserable acute illnesses, respiratory infections, severe conjunctivitis or pink eye, and gastroenteritis. They aren't usually fatal in healthy people, but they are incredibly efficient at making you feel absolutely terrible.
Then we have Hepadenoviridae, hepa obviously meaning liver.
Right, liver plus DNA, Hepadenoviridae. This is hepatitis B. And this virus is the MacGyver of the microbial world. Its DNA genome is incredibly weird. It's circular, but one of the strands is actually incomplete. It has a physical gap in it.
A gap, like it's broken.
Like a bridge that isn't completely finished being built. So when it enters the host cell, the very first thing it has to do is use host enzymes to repair that gap and make a full, perfect, double-stranded circle. And interestingly, the text highlights its unique replication cycle. It actually uses a reverse transcription step turning RNA back into DNA, which is usually something only certain RNA viruses do. It's a remarkably complex little machine.
Now we get to a really famous one, Herpes viridae.
The squatters. Herpes comes from a Greek word meaning creeping. They are large, enveloped viruses, but their absolute defining biological feature is latent infections. They never, ever leave.
That is a terrifying thought. Once you have a herpes virus, you have it for the lifespan of the host.
For your entire life. You get infected, you get sick, your immune system fights it off and clears it from your blood. But the virus retreats. It travels up your nerve fibers and physically sets up camp in your sensory ganglia near your spine or your brainstem. And it just goes to sleep. It waits.
Waits for what exactly?
A moment of weakness, stress, physical trauma, intense sunlight, or another illness. Anything that temporarily drops your immune defenses. Then the virus wakes up, travels back down that exact same nerve fiber, and causes a fresh lesion, a cold sore or a blister.
And this family includes a lot more than just cold sores, right?
Oh, it's a huge family. Herpes simplex 1 and 2, sure. But also varicella zoster, which causes chickenpox. If you had chickenpox as a kid, that virus is currently sitting inside your nerves, waiting to possibly emerge as shingles 50 years from now. It also includes cytomegalovirus, Epstein-Barr virus, which causes mono, and HHV8, which causes Kaposi sarcoma.
They are really playing the long game.
They absolutely are.
Okay, rounding out the DNA lineup, Poxviridae.
The tank. Pox viruses are massive. They have a complex, brick-shaped structure. They are so big you can actually see them with a good light microscope. You don't even need an electron microscope. Pox refers to a sac or vesicle. Smallpox is the most famous member here.
What makes them a tank other than size?
Most DNA viruses have to physically navigate into the nucleus of the host cell because that's where the cell keeps all its DNA copying enzymes. The virus needs to borrow them. But pox viruses are so huge, they pack all their own gear. They bring their own enzymes, so they don't even need to bother entering the nucleus. They just set up their own mobile command base right in the cytoplasm.
Okay, so that is the DNA squad. Now we switch over to section 4, the RNA lineup. If DNA viruses are the stable establishment, RNA viruses feel like the Wild West.
They are the renegades, without a doubt. RNA is chemically much less stable than DNA. It falls apart easier. And more importantly, the viral enzymes that copy RNA are famously terrible at proofreading their work. They make genetic mistakes constantly.
Which means endless mutations.
Constant, rapid mutation. This is exactly why we have to formulate a brand new flu shot every single year, but the polio vaccine you got as a child still protects you decades later. RNA viruses are shapeshifters.
Let's run through these families, starting with Picornaviridae.
Pico means small, plus RNA. Tiny RNA viruses. They're ether-resistant, which means they don't have an envelope, but do not let their tiny size fool you. This family includes the enteroviruses, like polio, which can paralyze you, and it also includes the rhinoviruses, which cause the common cold, also hepatitis A. So this one family can either give you the sniffles or alter your life forever.
Next is Calyciviridae.
Calyx means cup. Under the microscope, they have these cup-shaped depressions on their surface. The most famous member here is the norovirus, the cruise ship virus.
Oh, brutal.
It causes epidemic gastroenteritis. It is unbelievably contagious and very environmentally resistant. It hits hard, causes severe vomiting and diarrhea, and squeaks through closed populations rapidly.
Rheovirdae.
The weirdo of the RNA group. Rheo stands for respiratory enteric orphan. Here is why they are weird. Most RNA viruses have single-stranded RNA. Rheoviruses have double-stranded RNA.
Like DNA usually is.
Exactly, and their genome isn't just one long string, it is segmented. It's chopped up into 10 to 12 separate pieces. Rotavirus is the major player here, which is a major global cause of severe gastroenteritis in infants.
The outline lists arboviruses next, but it notes this is an ecological grouping, not a true genetic family.
Right. Arbovirus simply stands for arthropod-borne virus. It's a job description. These are viruses transmitted by insect vectors, primarily mosquitoes and ticks. They have incredibly complex life cycles involving both vertebrates and insects. The actual viruses in this group belong to several different families, but they share this transmission strategy.
And Togaviridae and Flaviviridae are often arboviruses, right?
Yes. Togaviridae, meaning cloak or envelope, includes rubella, which is not bug-borne, that's German measles, but it also includes several encephalitis viruses that are transmitted by mosquitoes. Flaviviridae is a very nasty group. Flavus means yellow, named for yellow fever. It includes dengue, West Nile, and Zika. And interestingly, it also includes hepatitis C, even though hep C is blood-borne and not spread by insects.
Next up, Coronaviridae. I think we're all pretty familiar with this one now.
We certainly are. Corona means crown, referring to those large petal-shaped peploma projections that look like a solar corona. They are large, enveloped, single-stranded RNA viruses. In fact, they have the largest genome of any true RNA virus. They cause common colds, but as we know from SARS, MERS, and COVID-19, they are highly prone to jumping species barriers from animals to humans with devastating effects.
Then we have Retroviridae, the absolute rule breakers.
Perhaps the most intensely studied family in modern virology. This is HIV and leukemia viruses. They are called retro because they work backward.
Breaking the central dogma of biology.
Exactly. The normal flow of genetic information is RNA makes protein. Retroviruses carry an RNA genome, but they pack a unique enzyme called reverse transcriptase.
They literally turn their RNA back into DNA.
Yes. They enter the cell, read their RNA, and write a DNA copy of themselves. Then they take that DNA and permanently splice it into the host cell's own chromosomes. Once that happens, the virus is literally a part of your genetic makeup. You cannot get rid of it without destroying the cell itself.
That is insidious. Okay, Erythemyxoviridae.
This is influenza. The key feature the text highlights here is the segmented genome. The flea virus doesn't store its genes on one strand. It stores them on 8 separate pieces of RNA.
Why is that a biological advantage?
Imagine 2 people shuffling 2 different decks of cards. If a bird flu virus and a human flu virus happen to infect the exact same pig cell, they both drop their genetic cards on the table. When it's time to assemble new viruses and pack up to leave, a new virion might accidentally grab 6 human cards and 2 bird cards.
Giving it a completely new hand.
Exactly. This is called genetic reassortment or an antigenic shift. It creates a hybrid virus that human immune systems have never seen before. This is the exact mechanism that sparks global flu pandemics.
The outline also mentions Paramyxoviridae. How are they different?
Para means closely resembling. They look and act a lot like the myxoviruses, the flu, but their genome is one single non-segmented piece of RNA, so they can't do that dramatic card shuffling trick. This group includes measles, mumps, and RSV.
Rhabdoviridae.
The bullet. Rhabdos means rod. If you look at them under an electron microscope, they look exactly like a bullet. The most famous member is rabies. It's a fiercely neurotropic virus that travels straight up the nerves to the brain, fatal almost 100% of the time when symptoms start.
And Filoviridae.
The thread, phylum, these are very long thread-like viruses. They often look like tangled shepherd's crooks or weird loop-de-loops under the microscope. This is Marburg and Ebola. They cause severe hemorrhagic fevers, highly lethal, causing widespread vascular leak and bleeding.
We also need to add Ariniviridae. and the Bunyaviridae groups.
Right. Arenoviridae arena means sand. When you look at them, they look sandy inside. That's actually host cell ribosomes that the virus accidentally packaged into its envelope. Lassa fever is the big one here.
Sure.
Bunyaviridae includes the hantaviruses, which are rodent-borne, and Bornaviridae is a very unique group of neurotropic viruses.
Okay, it is a total rogues gallery of horrors. But Section 5 of the text moves into something even stranger. The outliers. Things that aren't even quite viruses. Subviral agents.
We are definitely entering the biological twilight zone here. First, viroids.
Viroids.
These are strictly plant pathogens, as far as we know. They are incredibly small. They consist solely of a single, small, circular piece of RNA.
Okay, so what's missing?
No protein code, no capsid, no envelope. Just naked code floating around. It literally doesn't fit the fundamental definition of a virion.
So it's basically software without a hardware box.
Exactly, just raw instructions. And yet, this naked piece of code is perfectly capable of spreading and absolutely destroying entire fields of potato crops.
And the second outlier, prions.
This is the one that truly keeps biologists up at night. A prion is an infectious particle composed solely of protein.
Wait, just protein?
Just protein.
Yeah.
Absolutely no nucleic acid, no DNA, no RNA, no genetic blueprint whatsoever.
How can it possibly replicate or infect anything if it has no genes?
It does it through a conformational change. I call it the zombie protein theory. You actually have normal prion proteins naturally existing in your brain right now. They are folded into a nice, safe, functional shape. The infectious prion, the disease agent, is the exact same sequence of amino acids, but it is folded wrongly, like a tightly crumpled piece of paper instead of a flat sheet.
And what happens when the crumpled one touches the flat one?
It physically forces the flat one to crumple. It acts as a template. It's a domino effect. One bad protein corrupts its neighbor, which corrupts the next. Slowly, over years, your brain tissue turns into a literal sponge. This is mad cow disease and Creutzfeldt-Jakob disease in humans. It fundamentally proved to science that you do not need DNA or RNA to be infectious. You just need a corrupting shape. Furthermore, they are highly resistant to heat, UV, and formaldehyde. They are incredibly hard to destroy.
That is incredibly profound and genuinely disturbing. Let's try to bring some order back to this chaos and move to Section 6, architecture and chemistry. We talked about capsids earlier, but there's a specific geometry to this, right? The symmetry.
Nature absolutely loves efficiency. If you are a tiny virus and you want to build a sturdy protective shell using the fewest number of identical genetic instructions, meaning identical protein pieces, there is one 3D shape that wins every single time: the icosahedron.
Like a 20-sided die from a board game.
Exactly like that. The text calls this cubic symmetry. It has 20 identical faces, each one an equilateral triangle. It essentially approximates a sphere, but it's built out of rigid, flat structural plates. It has 5-3-2 rotational symmetry. Most viruses use this specific shape because it is structurally incredibly sound and biologically economical.
But there's another shape mentioned.
Right.
Helical.
Right. This is more like a spiral staircase. The protein subunits directly bind to the viral RNA and just wind around it, forming a long, continuous tube. And the text notes a very hard absolute rule here. Only RNA viruses can have helical symmetry. You will never see a helical DNA virus.
Oh, interesting.
And an even more specific rule for humans. All known animal viruses with helical symmetry are enveloped. You never see a naked exposed protein spiral causing a human infection. They are always wrapped in a stolen lipid cloak.
And the third type is just complex symmetry, which we mentioned with the massive pox viruses.
Right. They look like bricks with ridges. They don't follow the neat geometry.
Now, regarding the chemistry of these structures, the text breaks down the composition. Viral proteins, viral nucleic acids, lipids, and glycoproteins.
Yes. The viral proteins obviously make up the shell, protect the genome, and determine the antigenic characteristics, meaning what our immune system sees. The nucleic acids, the genome itself, can range vastly in size. Hepatinoviruses have a tiny genome, maybe 3.2 kilobase pairs, while poxviruses have massive ones up to 375 kilobase pairs.
And the lipids. The text mentions a very specific lab test regarding lipids.
This is a classic virology concept. Viral lipids are only found in the envelopes, and lipid membranes are highly sensitive to ether or chloroform. So if you isolate an unknown virus in the lab and you want to know if it has an envelope, you just treat the sample with ether.
And if it dies?
If it loses its infectivity, it means the ether dissolved its envelope. It's an enveloped virus. If it survives the ether unharmed, it's a naked virus. It's a very simple, definitive diagnostic test.
Let's actually stay in the lab. Section 7 covers cultivation and quantification. If I want to study these things, I can't just swab them onto an agar petri dish with some sugar like I would for strep throat, right?
No, absolutely not. That works beautifully for bacteria because bacteria can feed themselves. But remember, viruses are obligate intracellular parasites. They need a living host factory. You have to provide them with living cells.
So what do researchers actually do?
Historically, they used embryonated chicken eggs, and we actually still use eggs to grow the flu vaccine today. But modern virology relies on cell cultures. We take cells, maybe from a monkey kidney or human tissue, or often a human cancer tumor line, and we grow them in a plastic flask bathed in a nutrient broth. They grow until they form a monolayer, which is just a single sheet of cells like a carpet on the bottom of the flask.
And then you basically just dump the virus sample onto the carpet.
Right. But then you have a problem. How do you actually know the virus is there and multiplying? You can't see the virus particles with a standard microscope, so you don't look for the virus, you look for the crime scene. You look for the damage it leaves behind.
The cytopathic effects, TE.
Exactly. You look through the microscope at the cells themselves. Are the normally flat cells rounding up and detaching from the glass? Are they swelling with vacuoles? Are their membranes bursting? Or, as we mentioned earlier, are they fusing together into giant, multinucleated monster cells called syncytia?
The text also emphasizes inclusion bodies as a key diagnostic tool.
Those are visually fascinating. They are basically the physical virus factories inside the cell. The virus is running the cell's manufacturing machinery so incredibly hard that massive piles of newly minted viral proteins, or DNA, start accumulating in the cytoplasm or the nucleus.
Like a backlog of inventory.
Exactly. And you can see these dense globs with a simple chemical stain. In rabies infections, for instance, they form very specific blobs in the brain cells called Negri bodies. If a pathologist examines brain tissue and sees Negri bodies, that is a 100% confirmed diagnosis of rabies.
Okay, so we can see the damage, but how do we count them? If we need to know exactly how much virus is in a vial.
There are physical methods and biologic methods. Physical methods include PCR, which amplifies and counts the genetic material. But PCR has a flaw. It counts all viral DNA, even the defective, broken variants that can't infect anything. It doesn't tell you how many active, dangerous viruses you have. Electron microscopy is similar. You can physically count the particles on a grid, but you don't know if they are alive or dead.
So what's the biologic method?
The plaque assay. This is brilliant. Classic low-tech science. You grow your carpet of cells. You infect them with a highly diluted sample of the virus. Then you pour a thick, clear agarose gel over the top of the cells. The gel physically stops the new viruses from just floating away into the liquid.
Oh, it traps them locally.
Right. So when one original virus enters a cell, replicates, and bursts out, the new virions can only reach the immediately adjacent neighbor cells. They infect those, kill them, and move to the next circle of neighbors. It expands outward radially.
Like a crop circle in a field.
Perfect analogy. Eventually, you can look at the flask and see a clear, visible hole in the cell layer where all the cells are dead. That dead spot is called a plaque. And the math is simple. You count the holes in the lawn. If you have 50 plaques, you know you started with roughly 50 infectious viral particles in your sample. This gives you the infectious titer.
And the text introduces the concept of ID50 here as well.
Right, the infectious dose 50. This is the amount of virus required to successfully infect exactly 50% of the test hosts or tissue cultures. It's a standard measure of how potent or contagious a viral strain is.
All right, now we arrive at what I think is the most cinematic part of the whole episode, Section 8, the life cycle. The mechanics of replication. I really want to walk through this step by step. The text calls the very first phase the eclipse period.
This is genuinely the defining moment of virology.
Why? What makes it so special?
Because of what physically happens to the virus particle. Think about it. If a bacteria enters your bloodstream, it's still a bacteria. It swims around, it divides, it eats. But when a virion successfully enters a host cell, it completely vanishes.
It's just gone.
The virion, that beautifully structured geometric particle we just described, completely disassembles. It uncoats. It physically breaks its own shell apart to release the naked genome inside. If you were to take that cell and grind it up at that exact moment, you would not find any infectious virus particles. You would just find raw viral proteins and floating RNA. The virus has essentially sacrificed its physical body to release its code.
It's a suicide mission that leads to mass resurrection.
That is beautifully put. That period of invisibility is the eclipse phase. It's the time gap between the original virus dismantling itself and the very first newly assembled virus appearing in the cell. Infectivity is temporarily completely lost.
Let's trace the exact steps of this invasion. Step 1, attachment.
This is the lock and key mechanism. The virus is just floating blindly, it bumps into a cell, but it can't just enter any random cell. Its surface proteins, the keys, must perfectly match a specific receptor, the lock on the host cell's surface.
So it's highly specific.
Incredibly specific. This interaction strictly determines the host range and tissue tropism. This explains exactly why the influenza virus infects the cells lining your lungs, but ignores the cells in your big toe. The toe cells simply don't have the right lock on their surface.
Step 2, penetration and uncoating.
The actual breach. Once the key turns the lock, the cell is tricked into letting it in. The cell might actively swallow the virus in a bubble, that's endocytosis, or if it's an enveloped virus, it might just fuse its lipid coat directly with the cell membrane and dump the core inside. Once inside, the protein capsid opens up and the genome is released into the cytoplasm.
Step 3, synthesis, the hostile takeover.
This is where the virus takes the wheel. The viral genome issues orders that completely shut down the cell's normal production. It says, stop making skin cell proteins, start reading my code, and start manufacturing virus parts.
And the text makes a major distinction here between DNA and RNA viruses again regarding where this happens.
Yes, location is everything. Generally speaking, DNA viruses immediately head for the nucleus. Why? Because that's where the cell keeps its own DNA copying machinery. The virus goes into the nucleus to use the host's polymerase tools. Pox viruses are the lone exception here. As we said, they are massive and bring their own tools, so they stay out in the cytoplasm.
And what about the RNA viruses?
RNA viruses generally stay out in the cytoplasm, but they face a major biochemical hurdle. Your human cells do not possess a machine that reads RNA and copies it into more RNA. We only read DNA to make RNA. We don't go RNA to RNA.
So how do they replicate?
They have to bring or build their own custom machine. They code for a unique enzyme called an RNA-dependent RNA polymerase to do the copying work for them.
Step 4, assembly and release, the grand escape.
The factory has done its job. Millions of fresh copies of the viral genome and millions of new capsid proteins are floating around the cell, and then they self-assemble. It's almost like magic. The biochemical physics of the protein shapes just naturally snap them together around the genome. Once assembled, they have to get out.
And there are two main ways out, right?
Right. Lysis or budding. Lysis is the brute force, explosive method. The cell gets so physically stuffed with millions of new virions that the membrane simply ruptures. The cell dies instantly and the viral army floods out. This is very common for naked viruses like polio.
And budding?
The stealth exit. This is specifically for enveloped viruses. As we discussed, they gently push their way out through the host membrane, taking a little piece of it with them. The crucial difference here is that the host cell doesn't necessarily die immediately. It becomes a zombie factory, continuously shedding new viruses out into the body for days or weeks until it eventually burns out and dies of exhaustion.
That perfectly leads to the outcomes of infection. Productive infection is what you just described, right? Getting new viruses. But what about an abortive infection?
An abortive infection is a failure on the virus's part. The virus gets inside, but maybe the cell is the wrong type. The text calls it non-permissive. It lacks some specific cellular enzyme the virus needs to finish synthesis. The virus gets stuck, it eventually degrades, and the cell actually survives.
And latent infection.
The Cold War scenario, the virus gets in, the genome travels to the nucleus, but it doesn't initiate synthesis. It doesn't make any viral particles. It just integrates or sits there as a silent plasmid, just watching and waiting for the right moment to reactivate.
Moving to Section 9, which covers genetics and interactions. We discussed how RNA viruses mutate constantly because of bad proofreading, base pair changes. But the text mentions much larger genetic shifts through interactions between different viruses. We talked about reassortment, the shuffling of the genetic deck cards with the flu, but what is recombination?
Recombination mostly happens with DNA viruses. If two different but related viruses infect the exact same cell, their DNA strands can actually cross over and physically swap chunks of code. It's a literal splicing of two genomes into a new hybrid.
And then there's phenotypic mixing. I have to say, this sounds exactly like a trick from a spy novel.
It really is an espionage tactic. Imagine you have virus A and virus B, two different strains, infecting the same cell at the same time. The cell is chaotically manufacturing parts for both. Sometimes the genome of virus A accidentally gets stuffed inside the protein capsid shell of virus B.
A wolf wearing a different wolf's clothing.
Exactly. This newly mixed virion leaves the cell, On the outside, it has the shell and the spikes of virus B, so it can easily attach to and penetrate new cells that virus B normally targets. But once it gets inside that new cell and uncoats, it releases the genome of virus A.
So it breaks into a house using a stolen specific key, but once inside, it acts like the original burglar.
Yes, but here is the major catch. The genome inside is still pure virus A, so when it starts replicating, all of its offspring will be pure, normal virus A particles. The disguise only lasts for exactly one single generation. The text calls this a transcapsident.
That is fascinating. What about interference?
That's a turf war. Sometimes, if a cell is infected by one virus, that virus actively alters the cell or produces chemicals like interferon that completely block any other virus from infecting that same cell. It's essentially locking the door behind itself.
Let's zoom out now to the macro level. Section 10. Ecology, survival, and biothreats. How tough are these virions outside the body in the real world?
It varies wildly by family, but generally speaking, they are surprisingly fragile. Heat is a major enemy. For most viruses, being exposed to 56 degrees Celsius for about 30 minutes completely denatures their proteins and deactivates them. They essentially cook like an egg white turning solid. Polyomaviruses and hepatitis B are notable exceptions. They're surprisingly heat resistant.
But what about cold? Does winter kill them?
No, cold preserves them. Cold is their friend. If a lab wants to keep a virus viable for 50 years, they put it in a deep freeze. They are incredibly stable at negative 70 degrees Celsius or lower.
What about pH levels, acidity?
This acts as an evolutionary filter. Think about the enteroviruses like polio or hepatitis A. They are transmitted via the fecal-oral route. That means to infect you, you have to swallow them, and they have to pass through the extreme hydrochloric acid bath of your stomach to reach your intestines. So they have evolved to be highly acid stable. If they weren't, they'd go extinct.
Makes perfect sense.
But look at a rhinovirus, the common cold. It is transmitted via respiratory droplets. It only needs to survive in the mild environment of your nasal cavity. If you swallowed a rhinovirus, your stomach acid would instantly destroy it. It's acid label.
And antibiotics?
Zero effect. Antibiotics target cellular machinery like bacterial ribosomes or bacterial cell walls. Viruses don't have those structures. Giving antibiotics for a viral infection is biochemically useless.
Let's talk about how they move around. Transmission modes. We have direct person-to-person, indirect via fomites or fecal-oral, and animal-to-human zoonosis. But the outline specifically highlights the complex arthropod cycles.
Right, the insect vectors. This is fascinating ecological engineering. You essentially have two main loops. You have the urban cycle. This is human to mosquito to human. The mosquito is basically a dirty flying needle moving the virus between densely packed people. Dengue fever in a city operates like this.
And the other loop?
The sylvatic cycle. Sylvatic meaning jungle or forest. This loop goes from a wild vertebrate like a monkey to a jungle mosquito back to a monkey. It just sinkles silently in the wild population.
And the problem starts when humans wander into that jungle cycle.
Exactly. A logger or a farmer goes into the forest, gets bitten by the infected jungle mosquito, and brings the virus back to the city, igniting an urban cycle. Yellow fever does exactly this.
And the text also introduced the concept of a dead-end host.
This is a crucial concept, perfectly illustrated by West Nile virus. West Nile normally cycles constantly between wild birds and Culex mosquitoes. A mosquito bites an infected bird, picks up the virus, and then bites a human. The human gets very sick.
The cycle stops there.
Right. Humans don't replicate the virus to high enough concentrations in our blood to infect a new mosquito if it bites us. So the chain is broken, we suffer the disease, but we are an epidemiological cul-de-sac, a dead end.
The chapter concludes with a really sobering look at emerging diseases and biothreats. Why are we seeing so many new viruses popping up?
The text lists several major drivers. Environmental change and deforestation are massive ones. We are pushing deep into pristine habitats and coming into contact with wildlife and their unique viruses that we simply shouldn't be interacting with.
Right.
And air travel. A virus that emerges in a remote jungle village used to just burn out locally. Now it can be on a flight to London or New York in 12 hours. Viruses effectively have passports now.
But the text also lists technology as a driver. That surprised me. How does technology drive viral emergence?
Specifically xenotransplantation.
Meaning using animal organs in humans.
Yes. We have a massive shortage of human organs for transplants. So medical science is aggressively experimenting with putting genetically modified pig hearts or pig kidneys into humans. It's a miracle of surgery, but it carries a hidden virological risk.
What's the risk?
Pigs carry their own latent viruses. specifically porcine endogenous retroviruses, deeply embedded in their DNA. If we transplant a living pig organ into a human whose immune system is intentionally suppressed to accept the organ, we are giving that pig virus an unprecedented opportunity. If that animal virus learns how to efficiently infect human cells, mutate, and then spread from that patient to the public,
We could literally manufacture our own novel pandemic in a surgical suite.
exactly, we are forcefully crossing a biological barrier that normally takes evolution millennia to cross.
Wow. And finally, regarding bioterrorism, the text specifically flags smallpox and the hemorrhagic fevers, like Ebola and Marburg, as the big guns, the ultimate threats.
And if you think about their viral properties, it's obvious why. Smallpox is highly stable in the environment, it transmits easily through the air, and it has a high mortality rate in a population that is no longer vaccinated against it. Ebola is terrifyingly lethal, though harder to spread. The absolute nightmare scenario for biosecurity is someone using genetic engineering to combine those traits. If you could engineer a virus that had the airborne transmissibility of smallpox, but the hemorrhagic lethality of Ebola, that is a civilization-ending weapon.
We have covered an absolutely massive amount of ground today, all the way from a 20-nanometer inert virion crystal, through the mechanics of hijacking a cell, to the complex ecology of a global pandemic threat.
It's a vast subject, and we really just scratched the surface of the underlying molecular biology.
But the main takeaway for me, looking at all this source material, is the sheer efficiency of it all. Viruses aren't evil. They aren't malicious. They are just code. They are a ruthless biological algorithm that simply says, copy me. And they are better at executing that algorithm than almost anything else on Earth.
I agree completely. They strip life down to the absolute bare basics. A geometric shell and a whisper of genetic instruction. And yet, they dictate the survival of entire species.
And in doing so, they actually reveal so much about how our own cells work. You really can't understand human biology without intimately understanding the microscopic things that evolved to hijack it.
Exactly. They're the ultimate teachers of molecular biology, if we pay attention.
A sobering but incredibly fascinating deep dive. Thank you so much for guiding us through this invisible world today.
It was a real pleasure. Thanks for digging into the details with me.
And to you listening, I want to leave you with one final thought to mull over. We talked about how retroviruses permanently integrate into our DNA and how we are experimenting with animal organs. Given that roughly 8% of the human genome is actually made up of ancient, deactivated viral DNA from infections millions of years ago, if viruses actively shape our evolution, and we are now engineering viruses and mixing species, are we essentially taking the wheel and engineering the next bizarre step of human evolution through viral integration? Keep your immune system strong, wash your hands, and stay curious. We will see you on the next deep dive.